Here we go.
Late night show.
Okay, hello everyone. Welcome to the last talk of the career fair, which has been rescheduled to today's session due to some technical issues yesterday. And it is my great pleasure to be joined by Dr. Spencer Wells. To me, Spencer is a very impressive personality. He has started his academic career with a PhD at Harvard University, which was followed by a postdoc at Stanford University working on human genetics. Since then, he has been a key player in mapping human migration history using genomics. While serving as National Geographic's Explorer in Residence, he has visited more than 100 countries, collecting DNA samples of indigenous populations from around the world as part of the Genographic project. In recent years, he has moved into entrepreneurship with the founding of the personal genomics company Incitome. Incitome has now been transformed into the non-profit, the Incitome Institute, focusing on genomic identity and privacy. Spencer has also been part of the production of documentaries, has written several books, and is the co-owner of a blues club in Texas. Spencer, thank you for being here with us at the rescheduled time, and we are very, very, very much looking forward to your talk.
Thank you, Barbara. I hope everyone can hear me, and I apologize for the technical issues from last night. I am tuning in from Indonesia, where I now live, so slightly different from Austin, Texas. That's the view right now. You're literally looking at Wallace's line there. So those of you who are students of evolution will know about Alfred Russel Wallace and the work that he did in the Malay Archipelago, in other words, Indonesia, deciphering biogeographical lines in the region and discovering many new species. So it's a fascinating place to be, but it's great to be here to be able to talk to you about the work that I've done. I'd like to leave some room at the end because a lot of my time over the last few months has been devoted to commenting on coronavirus and coronavirus policy in particular, most notably in my country of birth, which has not done a particularly good job with the coronavirus, to say the least. And so I'd be happy to answer questions about that. But anyway, let's kick it off. So, the human journey, genetic odyssey. Well, so I'm a human population geneticist by training. As Barbara said, I got my PhD with Dick Lewontin, Richard Lewontin at Harvard, in what was arguably the greatest evolutionary biology department that has ever existed. So Steve Gould and Ed Wilson and Ernst Mayr and others were all teaching and came to lab meetings. It was an intimidating environment to cut your teeth as a young academic. But although I started off studying the basic mechanisms of evolutionary biology, I soon decided that I wanted to apply them to the study of human variation. And humans are an incredibly diverse species. There's 7.7 billion of us spread around the planet today, in a dizzying array of skin colors and hair types and heights and all sorts of other things that serve to distinguish us phenotypically on the surface. And really, one of our primary goals as human population geneticists is to explain these incredible patterns of human diversity around the world. How did they arise, and how did we arrive at the patterns that we see today? So moving forward. Now, as a scientist, as someone who really tries to kind of cut to the marrow of the problem and really get at the key questions, what we're really talking about here are questions of origins and journey. Where did we originate as a species, and when? Was it millions of years ago? Was it in multiple places around the world, which is something that was widely accepted until relatively recently? Or was it a single point of origin more recently than we might have suspected? And if we do originate from a common origin, how did we occupy every corner of the globe? The question of journey. And so, these are really the key questions we're getting at using the tools of science. Well, so trying to answer these questions of human origins and journey, it's really something that has been the purview of the field of paleoanthropology for over a century, nearly a century and a half now, if you go back to the discovery of the Neanderthal type specimen in the 1850s. Digging things up out of the ground and guessing at how they may or may not connect to things in the present. What you're looking at here, for instance, are 3 potential human ancestors. But interestingly, all uncovered from the same location in northern Kenya, on the shores of Lake Turkana by the Leakey family, and all dated to roughly the same time, about 2 million years ago, from left to right Homo erectus, Homo habilis, and Paranthropus boisei, a robust Australopithecine. So which one of these are we actually descended from? It's a deep question. I mean, they all lived at the same time, so we can't be descended from all 3, but they're all potential ancestors. Well, so getting at the answer to that question is a little bit more complicated, and it involves moving from stones and bones to a genealogical approach. So as a person trained in genetics with a PhD in population genetics and, the harder sciences, if you will, I'm a data freak, as I'm sure many of you in the audience are as well. I mean, we use large quantities of data to try and answer very complicated questions. So instead of digging up, something out of the ground, which, may be completely new, but it's N of 1, and guessing at how it may connect to the present and into our family tree, we as geneticists gather large quantities of data, mostly from people alive today, but, and I'll get to this toward the end of the talk, also from ancient material buried in the ground for thousands, tens of thousands, even potentially hundreds of thousands of years in some cases. And we take a slightly different approach. Instead of guessing at how these ancient materials may connect up with the present, we start in the present and work our way back in time. It's a genealogical approach because we know with absolute certainty that every person alive today had parents, and those parents had parents, and so on. So it should, in principle, be possible to build a family tree for everyone on Earth today. It's a genealogical approach. Now, anyone who has built a family tree, who's asked their parents and grandparents about where we all came from and the villages that they originated in, knows that the written record runs out eventually. But of course, we have another written record, what is in essence a history book written inside of our DNA that we carry in nearly every cell in our body. And that is, the genetic material that we use to track these ancient human migrations. Now, you're all scientists, so I don't have to show you this, but, just a quick reminder that the human genome is massive. It's billions of nucleotides and linked 3 billion in a haploid human genome. And in every generation, we have to copy all of those billions of nucleotides through the process of DNA replication. Now, when this happens, although there are proofreading mechanisms built in, of course, on occasion, you're going to have a misincorporation of a nucleotide. Substitute a C for a G or an A for a T or vice versa. These mutations, as they get passed down to children and grandchildren and so on, become markers of descent, such that if you're carrying a certain pattern of genetic markers, we can trace you back in time on a particular genetic lineage and ultimately create what is in effect a family tree for everyone alive today. And starting back in the 1990s with mitochondrial DNA on the left, the famous story of mitochondrial Eve, Alan Wilson's group, Becky Ken and others in his group, analyzing mitochondrial DNA, discovered that everybody alive today likely traces back to an African origin within the last 200,000 years. The Y chromosome work was work that we really pioneered in Luca Cavalli-Sforza's group at Stanford in the 1990s when I was a postdoc there. And it traces back and tells a very similar story. We all trace back to an African origin, the longest branches in the human family tree. And because, of course, the branch length is determined by the number of mutations, the amount of time that these branches, these lineages have been accumulating changes in their DNA through history. The longest branches are found only in Africa. What that tells us is that Africans have been accumulating mutational diversity for longer, and therefore our species originated in Africa. Now, Even Darwin, speculated on this in not The Origin of Species, his most famous book, but in The Descent of Man, published in 1871, I believe, he said it's likely that humans originated in Africa because there are chimpanzees and gorillas and things that look very much like us there, but he had no data for it. This is kind of coming full circle. It's confirming what Darwin said about an African origin, but it's changing the date substantially because apes first appear in the African fossil record. And yes, they did originate. Apes originated in Africa with proconsul about 23 million years ago in the northern part of Africa, at a time when it was disconnected due to plate tectonics from the rest of the world's land masses. And it bumped into Eurasia between 16 and 18 million years ago. That's when the ancestors of the gibbons and the orangutans moved out into Eurasia. So yes, absolutely. If you take a Darwinian view, going back to the latter part of the 19th century, we clearly originated in Africa, but it was tens of millions of years ago. What the genetic data adds is the recency of that date. And this is what was so astounding. When mitochondrial Eve was first discussed, first published, in a series of papers in the late 80s, early 1990s, picked up by the popular press. And then, of course, the Y chromosome dates, much more recent than that with our initial estimates, but now coming back to that 150 to 200,000 year date. That's very, very recent in an evolutionary timescale. And particularly when you start to look at the lineages that appear outside of Africa, we're talking events that have only happened in the last 50 to 60,000 years. And so we have a very recent common origin in the African continent, and then a rapid spread out of Africa within the last 60,000 years or so, spreading along the southern coastline of Asia, reaching where I am today, Indonesia and Australia, by at least 50,000 years ago, probably even earlier than that. And inland migration, moving up through the Middle East into Central Asia, crossing westward into Europe and eastward into the Americas by 20,000 years ago. These are the incredible Paleolithic journeys of our species. And this is what I've spent my career trying to understand, as well as more recent events, which we'll talk about in a minute. Now, that was the clean, tidy story. Circa, say, 15 years ago, when we began the Genographic project at National Geographic, so around 2005 when we launched that project. And there have been two significant revolutions since then in certainly population genetics, but I think in genomics more broadly. The first one, of course, is our ability to sequence whole genomes. I mean, I took my first genetics class, and it was very much genetics. We were doing Punnett squares, and it was basically agricultural genetics, a little bit of quantitative genetics, but very, very simplistic compared to modern genomics. I took that first class in 1986, and DNA sequencing had just been invented a few years before. Wally Gilbert and Fred Sanger, for their respective methods of learning how to sequence DNA, had just won the Nobel Prize a few years before. And the concept of sequencing entire human genomes was just magical. It was, unimaginably difficult. And the work that I did during my PhD using manual sequencing, literally using a ruler to track up through the A, C, G, and T lanes on an autoradiograph and type this in by hand. I mean, you're limited to sequencing a few hundred nucleotides a day. And so people were doing calculations around the time I started graduate school in 1989 that, would literally take centuries to sequence a human genome. And of course, that has all changed significantly, with the advent, of course, of automated sequencing in the 1990s, which led to the completion of the first Human Genome Project, literally a brute force effort. But really, the big shift was in 2007 with the advent of next generation sequencing and the ability to sequence whole genomes literally in a day for $1,000 or so, the current price point. Could go even lower if Illumina decides to lower its prices, or if Oxford Nanopore comes up with a better solution. But the point is, This is technology that was unimaginable when I started in the field of genomics, and it's opened up the field of population genetics to amazing new discoveries, making use not just of those uniparental markers, which we had studied for so long, mitochondrial DNA and the Y chromosome, which we were limited to studying mutational diversity, estimates of the mutation rate, giving us ages of the lineages and the timings of these population splits as humans moved across the earth. We actually, I have to say, now that we're getting some more recent data, I feel like we did a pretty decent job of it. But still, it's nothing like the level of insight that you can gain from sequencing a whole genome. And we can now make use, of course, of the entire genome. And the genomes of thousands, hundreds of thousands, even millions of people to begin to gain new insights into the ebb and flow of human evolutionary history. And, much of the work that we did in Cavalli Sforza's group, work that I've done throughout much of my career, has been focused on migrational patterns. And so DNA is simply a piece of baggage that you take along with you. And it mutates as you have kids and grandkids and so on. And so we can track these breadcrumbs, mutational breadcrumbs, if you will, back in time and connect people into lineages. But the genome is not simply a passive piece of baggage that we carry with us. It's also adapting actively. And much of the fascination in the study of human evolution is in how humans have changed over time. If we originated relatively recently in tropical Africa, which I think is incontrovertible mostly, although there's complications that I'll get to again in a minute with the ancient DNA work, but effectively we blasted out of tropical Africa within the last 2,000 generations. How have we adapted so rapidly to these different environments? And we can actually make use of some of the other aspects of genetic inheritance, including recombination patterns, to give us insights into that. And so if you think about chromosomes recombining down through the generations and establishing a baseline level of what we call linkage disequilibrium, so the relative lengths of these non-recombining haplotypes along the chromosomes in different populations, which are kind of population specific, related to population size, population history, admixture, and so on. But in effect, you have a kind of null background expectation of the pattern of recombination in modern human populations. And that allows you to go in and look for anomalies. And so if we think of this as, different colored beads on a string, these are different haplotype blocks along human chromosomes. And we expect a kind of random distribution among different chromosomes, but we find, in this case, a particular pattern that is far too long and far too widespread to be explained by chance. That's an indication that something has happened there. It's like, I've compared this in some of my popular talks and in a book I wrote to, a fashion accessory. So imagine, literally a necklace with different colored beads on it. And there's one particular pattern of beads, in this case, the blue, the green, the red, that's far too common in the population to explain by chance. Well, in fashion, that would be called a fad, or, something that is very popular and everyone wants it on their necklace. In biology, that's a sign that there is something there that is biologically important. There's been a selective event. And so we can actually scan the entire human genome in many, many people and see clear evidence of these selective events. The one that is at highest frequency is over here on the left, LCT lactase. So the lactase gene, mutation that allows people to digest lactose, the sugar in milk, the disaccharide in milk, into adulthood. All babies are born, all mammalian babies are born with the ability to digest the sugar, and most children lose that ability around the time they're weaned. So in the case of humans, that's around 2 years or so. And so the normal form in modern humans is that we are lactose intolerant. But in some populations, there's been a mutation near the lactase gene or in the lactase gene, depending on which population you're talking about. And these are the populations that domesticated sheep and cattle and goats. And so they have effectively figured out a biological way to continue to digest this biologically, diet important sugar into adulthood. And it's risen in frequency rapidly in certain populations, particularly in Northern Europe. And so literally within the last few thousand years, the lactase persistence gene or variant has gone to very, very high frequency in Northern European populations, 95 to 100% in say, Scandinavia. Whereas, again, the normal human form is to lose that ability to digest milk into adulthood. So it's a fascinating story, but there are literally hundreds of these little stories. Toll-like receptors, probably having to do with adaptation to viral pandemics, something which is very relevant right now. SLC45A2, which is light skin, light hair color, mostly found in Northern European populations. MHC, again, important in the immune response. FADS, association with the ability to digest fatty acids. Each one of these tells a fascinating story about how humans have adapted to the world as they've moved out of Africa and populated the rest of the planet. And this is really kind of the cutting edge of human evolutionary biology right now, in my opinion. It's really understanding, how we've adapted to high altitude in Tibet, for instance, by taking on a piece of DNA from one of our cousins. This is the other revolution that has occurred. The study of ancient DNA, our ability to obtain sequenceable genomes. And so we're not just talking about mitochondrial DNA again. We've been sequencing ancient mitochondrial DNA since the 90s, but our ability to sequence entire genomes, that's something that had to wait for next generation sequencing. And of course, many of you have probably tested yourselves through one of the companies, 23andMe, or perhaps Ancestry, or even Genographic, and discovered that you have a certain percentage Neanderthal DNA. Non-Africans have around 2% of their genome that has been absorbed from the Neanderthal population. So we didn't simply drive them to extinction. We actually mated with them and had offspring, and those offspring were incorporated into the human population. And today, around 2% of the average non-African's genome is Neanderthal in origin. There are also completely new species that have been discovered. Denisovans, originally uncovered from a pinky bone from a young girl and a tooth in a cave in Siberia, the Denisova cave. But probably their center of diversity was actually right down here in Indonesia, where there were multiple Denisovan mixture events. And we see the highest frequency of Denisovan DNA in New Guineans and Eastern Indonesians, 5%, 6% in some cases of their genomes coming from a local Denisovan population, probably in multiple waves of admixture. And so these insights from ancient DNA are fascinating. They're also kind of fun. And this is part of the reason why they, the companies that offer the DNA testing have had fun with the Neanderthal percentage. Ozzy Osbourne, of course, perhaps not unexpectedly, a little bit higher in Neanderthal DNA. But I want to end off by talking a little bit about the fact that this is not simply limited to the earliest days of our species and the initial migrations out of Africa. Ancient DNA is really providing us tremendous insights into the ebb and flow of what we might think of as recorded history. Writing was invented at least 5,000 years ago in certain parts of the Middle East, Mesopotamia in particular, and in Egypt. And the Bronze Age really started to spread around that time. But much of what we knew about the Bronze Age was limited to these historical accounts, which were often somewhat mythological, and people didn't necessarily believe the stories of, massive slaughters and so on. And, this has been a strain in sociology and historical research for the last century or so. A rejection of a migrationist model and a replacement model of cultural shifts. But what ancient DNA is actively turning up is that in many cases, these shifts in culture, in particular with the spread of the Bronze Age into Europe in the last 5,000 years, have been the result of the massive migration of human populations. And so if we look at the coalescence point, and this is the big phylogenetic tree you're looking at here, these are all the Y chromosomes that you see in modern European populations. They all effectively coalesce back, 95% of them, to the Bronze Age. And, the Bronze Age was a relatively bloody time to be alive. It was pretty scary. There were slaughters that were carried out. The Bell Beaker people who moved into Britain around 2500 BC effectively replaced, to the degree of about 90%, the people who were living there before. And so, what we like to say is kind of a simple shorthand to help people understand it is the people who built Stonehenge toward the latter part of the 4th millennium BC were not the same people who were worshiping at Stonehenge in the middle part of the 3rd millennium BC. There was a population replacement from the steppes of southern Russia and Central Asia. And so, the stories that are being revealed are not necessarily the nicest aspects of human history, but they're fascinating nonetheless. And I want to go ahead and end it there and take some questions. And I have a podcast with Razib Khan called The Insight. You can get it on most of the major podcasting platforms if you're interested in these sorts of stories. We talk about recent findings in population genetics. Ancient DNA is a big focus of what we talk about. You follow me on Twitter and Facebook and so on. But thank you very much, and I'll be happy to take some questions. So, okay, let's go back to top. Sarah Jamis, I don't know if I'm supposed to say the names of the people asking questions, but how much does genetic drift account for human evolution in modern life in absence of strong selective factors? I think drift is a major, it's almost certainly the most important force. If you think about human populations moving around the world during the Paleolithic, these would have been small hunter-gatherer bands up until about 7,000, 8,000, 9,000 years ago. Dawn of the Neolithic, we typically say was around 10,000 years ago in most parts of the world, but it was limited to particular groups. But effectively, everybody's living like a hunter-gatherer, and hunter-gatherer groups, do not really get much larger than a few dozen people. An extended band might be, Dunbar's number of roughly 150 people, but we're talking relatively small groups. And so drift was a really important part of this. The adaptation, I think, largely comes in later, especially post-agriculture. As the population size increased, selective forces are much more important in larger populations. And so that's when we start to see, I believe, the effects of adaptation, and sexual selection and so on. Can genetic data help decipher the evolution of languages? Yes, we believe that it can, except in cases where it can't, in which case it also provides some insights. So, for instance, I've done a lot of my field work in Central Asia, Middle East and Central Asia. That's really been kind of the core focus of my work, although I've worked all over the world: Sub-Saharan Africa, North Africa, Southeast Asia, India. But my biggest expeditions in terms of sampling and the work that has dominated what I do as a scientist really focused on Central Asia. And so this is the land of the Turkic peoples. So the Uzbeks and the Kazakhs and the Turkmen, who did not originate in Turkey. They arrived in Turkey relatively recently, in the last 1,000 years. And so, one of the things that you can look at there, for instance, is what do the genetic patterns tell you about the spread of the Turkic languages, which, based on diversity patterns and so on, certainly originated in the Altai, or the western region of Mongolia. So they spread from that part of southern Siberia or western Mongolia down through Central Asia and into Anatolia over the last couple of 1,000 years. Well, the surprise is, even though the language that gives the family its name, so Turkic languages, is, the one that we think of as being the dominant language. In fact, the people living in Anatolia have a tiny percentage of Central Asian DNA. So around 5% or so. And so they are mostly, genetically related to people who were living there prior to the arrival of the Turkic-speaking peoples. And so that gives you an insight into the way in which these languages might have spread. And this goes back to work that was, done by Colin Renfrew and others back in the 1980s and the 1990s. But Renfrew suggested a model in which certain societies have some sort of technological advantage. He called it elite dominance that allowed them to come in and in relatively small numbers conquer a region without having a huge genetic impact. We see the same thing in Hungary. The Hungarian language is a Uralic language. It's not related to Czech or Slovak or Polish or any of the surrounding Indo-European languages in that part of Central Europe. It is closest to languages spoken over in the Ural Mountains in Siberia, in Western Siberia. And that's because of a migration, again, about 1,000 years ago from that region. Horsemen, the Magyars or the Huns, as some people would call them, arriving in present-day Hungary, and they conquered the region and imposed their language and their culture without a huge genetic impact. And so, knowing something about the distribution and the history and the relationships of the languages and the timing of those events based on historical records and archaeology, and then looking at the genetics allows us to put together the details of how these events might have played out. And so I think genetics, not completely on its own, but in concert with these other areas of study, allows us to say quite a bit about human history. What are the ethical concerns around personal genomic data? There are lots of ethical concerns, and we really don't have a lot of time to go into this now because I think we're coming up against the timeout period. But no, I mean, the biggest ethical concern for me is it's part of a broader issue of data privacy. It's something that the European nations, I think, have approached head on. And, thank goodness. I'm very pleased with, a lot of the work that has come out of Europe on data privacy and some of the legislative, legislation that has been passed. But in the United States, unfortunately, which is, kind of the Wild West, where a lot of these companies have been collecting data for decades now, to individual companies, and it's settled by lawsuits. And unfortunately, this extends into the genomic data realm, which is not necessarily scarier, but potentially scarier, depending on the complexity of the data, the granularity of the data, and how you use it. I mean, what really concerns me in the US's handling of personal genomic data is how it has been used by law enforcement to catch criminals. And I think most of us would agree that serial killers are not good people, and they should be put in prison and removed from society. But I think it's a very fine line, particularly with the administration that we have now in the United States, which I'm not a huge fan of. But the concerns are that they might start to use this data for other reasons. Profiling people, and who knows what they're going to do with it. But the point is that I feel like personal genomic data, like any other highly personal data, people should be informed of how it's going to be used. You should be able to opt out of it. We're actually in the process of preparing a white paper on this, which has been somewhat sidelined by the coronavirus. Because I think everybody scientifically is very focused on that, justifiably right now. But, eventually we're going to get past the pandemic, and we're going to get to the point where, personal genomics is again, a very important issue. How do you think our genomes will record the coronavirus pandemic? I don't know. I mean, it seemed pretty clear to me early on that there was a lot of interpersonal variation, interindividual variation in the response to a coronavirus infection. So some people are infected, and age is a major factor. So if you're young and healthy and female and not obese, in all likelihood, you're barely going to notice you had it. It's going to be like a bad cold. If you are older and male and obese and have type 2 diabetes, like many people in the Western world, and particularly in the United States do, you could end up in hospital on a respirator for months and have long-term effects. I don't see us adapting at this stage right now, because adaptation really means that the people who are most susceptible and have the highest morbidity and mortality literally go away, and so their genes are removed, and the ones who are best able to cope with it are the ones who survive and reproduce. I don't see it having that kind of level of selection right now, thank goodness. But no, there are definitely genetic factors that play into this. And, I was just reading earlier today that there's been a lot of debate about, blood type and the extent to which having a particular blood type predisposes to worse morbidity. 23andMe did a large-scale study, GWAS study of over 1 million people from their database, where they had collected information on COVID status, and there does seem to be something to the blood type. There are almost certainly, other genetic variants that have to do with, the response to this. But I don't see right now, I would say, that there is a significant level of genetic selection in this. I mostly see that there are, effects from these other lifestyle factors, i.e. age, obesity, and so on. But, that could change moving forward. We're in the early stages of a pandemic. And, this is one of the things that I've been trying to get across to people for months, is that this is not something that is simply going to blow over by April, which I think a lot of people in the US were hoping early on. It's not something that's going to blow over in the summer. It's not something that's going to blow over by this winter. If we're lucky, we might have a series of effective vaccines by early next year, distributed to mostly rich Western nations who can afford to pay for them. But I mean, this is something that's going to stick around for a while, and those vaccines are not going to be 100% effective in all likelihood. I'd be very surprised. I think they're going to be closer to a flu shot, where you're talking 50, 60, possibly 70% effectiveness. And then you have to factor in people's willingness to actually get a vaccination, which these days is certainly not 100%. And so we live in interesting times. Being a scientist in the 2020s is, it's fascinating, because you not only have to communicate your scientific findings to the broader public, but you actually have to make a case for why science is believable and important, which is not something I've experienced before really in my life. Although a decade ago, I wrote a book that included a fair amount on climate change, and I had a lot of pushback on that, but it was still relatively early days. We seem to be going into a very anti-scientific period in certain parts of the world, certainly in the United States, which concerns me greatly, because science is the only thing that's going to get us out of this pandemic. You have to trust in science, and you have to be honest about the data. And politicians who refuse to do that are, endangering their entire populations. So enough on that. We won't go any further into the politics. Are there any other ancient human species that have not even been uncovered yet? There are so many. We still don't have a clear picture of what Denisovans look like. And I actually think that Denisovans, again, going back to the ancient DNA, discovered from this cave in Siberia, but mostly an East and particularly Southeast Asian species, where, again, 5%, 6% of the genome of, peoples in Eastern Indonesia and New Guinea and Australian Aborigines are Denisovan in origin. I mean, this is a species we didn't even know about until a decade ago, and we only discovered it genomically. I think there are tons of undiscovered species right here in Indonesia, where I am now. And in fact, I'm going to be turning my eye to work that's a little bit more toward the paleoanthropology side, in an effort to try and find some of these creatures like the hobbits, the Homo floresiensis that lived a few hundred miles to the east of here on the island of Flores. I think there are probably undiscovered species from Sulawesi. We know that there are recently discovered species from the Philippines. All of these islands have been kind of a crucible for hominin evolution. I think there are a ton of species to be discovered. I think we'll discover new things in India and certainly in Africa as well. I mean, when we look at the genomes, the ancient DNA from Africa, and this is very early days, and we don't have samples that are that old that have intact DNA. But even looking at samples that are 5,000 to 10,000 years old, we see clear evidence of interbreeding with other things, other hominins that were there that are no longer there. And you really have to start to modify your view of human evolution. It is not a simple tree, like I showed you at the beginning. That's the main story. That's about 90% of it. But the other 10% is really fascinating and really complicated, and it may have resulted in a lot of biological adaptations, phenotypes that we carry with us to the present day that we took on from these other creatures that were human-like but weren't modern humans. So, I think there's a ton of work that still needs to be done in this area. Barbara, how are we doing on time?
I think we might have just about time for one more question.
Okay. What would be your suggestion to someone who is an aspiring evolutionary biologist, specifically interested in behavioral sciences, in terms of the kind of career path to take? That's a tricky one. So the field of behavioral genetics was very popular back when there was very little data. So we go back nearly a century, and there were a lot of people speculating on the genes for intelligence and other aspects of human behavior. And there was a huge backlash, of course, with the Nazi eugenics studies and the crimes that were committed in the Holocaust and so on, in the middle part of the 20th century. And there was a huge backlash in general against looking for a genetic basis for human behavior. That has started to come full circle. And there is now a great amount of work that's being done with huge cohorts, so 1 million or more people, using proxies for IQ, such as educational attainment, and other things. And there are genetic components to these human behaviors or behavioral attributes. It is nowhere near as simple as people once suspected it might be. These are all highly quantitative traits, and genes explain a tiny fraction of the variance. But I think, with our ability to study millions of human genomes, we're going to discover some of these very subtle effects, none of which will be deterministic. This is the important thing to remember. Human behavior is far too complicated to be reduced to a handful of SNPs. But, a collection of SNPs may have an influence on how behaviors develop in concert with the environment that individuals are brought up in. And I see this as a really exciting, new field of research that's really only been possible in the last 5 years or so. Stuart Ritchie, who is University of London, is doing some fantastic work on this, and there are others in the field as well. But I think it's utterly fascinating that we are beginning to pick up some of these, genetic components. Again, these are not determined by genes, but they're influenced by genes, to very complex human behaviors. And I'm a parent. I have 2 daughters, and any parent will tell you that the children that you have are not born as blank slates. They have certain predispositions when they are born, and you see those relatively soon in their lives. And yes, some of that is certainly due to environment, but some of it is almost certainly due to genetic factors as well. And so, again, we're not talking about biological determinism here, but we're talking about the ability of large-scale, high-throughput, genome-wide studies to yield some insights into, patterns of human behavior, things that may influence human behaviors that may be important as well. They may be important socially. I think the blank slate, the concept of everyone is born as a complete blank slate, and there are no genetic factors that play into human behavior and so on. I think that's good in some ways, but I think it's also bad because it effectively expects everybody to be the same. And as I said, anyone who's had children knows that, some kids are born as introverts and some are born as extroverts. And if you expect them all to behave as extroverts and become salespeople, or whatever it might be, you're really losing some of the nuances of human variation. And so I think understanding these things about ourselves is really interesting. And I think it's important for society.
Okay, Spencer, thank you. I think we are going to have to end it there because we are running a bit late for the next talk. But thank you very much for taking such great care to answer all the questions and giving us, touching on many other interesting topics. And thank you for the very insightful talk on our all history. So
Thanks,
we really
Barbara.
enjoyed it. Thank you for your time. And again, we really appreciated that you could make it today.
Absolutely. Bye, everybody.
Okay. Bye. See you in the next talk, which is by it's in the sticky note on the top of the chat. It's Abdul Rachid Tayam. So
Okay.
see you there. Goodbye.
Bye.